Let's subscribe this channel first for the biology session. DNA replication. So DNA replication is one of the most crucial processes in all living cells. It ensures that genetic information is accurately copied so that new cells inherit the same DNA. To make this easy, imagine DNA as a book of instructions that needs to be duplicated every time a cell divides. So the introduction and opening DNA. DNA double helix inside the nucleus. We start by introducing the DNA itself. The double helix is shown as a smiling, friendly character inside the cell's nucleus. This sets the stage. DNA is the genetic blueprint of life. The text DNA, the genetic blueprint, highlights its importance. Next, replication begins. Helicase, depicted as a cute robot or zipper operator, unwinds the two DNA strands, breaking hydrogen bonds. That replication fork formation. The Y-shaped replication fork appears next. This is where the DNA strands separate and new strands start forming. So the text replication fork formation emphasizes this structure. Origin of replication highlighted. The replication origin is the specific starting point on the DNA, where helicase begins unzipping. Highlighting this visually shows students where replication begins. So the text is origin of replication. Free nucleotides floating. To build new DNA, the cell needs nucleotides. They are depicted as floating characters ready to pair with the template strand. Text: free nucleotides available. Base pairing rules. Adenine pairs with thymine. Adenine A pairs with thymine T through two hydrogen bonds. Cartoon characters with glowing bonds make this visual memorable. Guanine pairs with cytosine. G pairs with C via three hydrogen bonds. The cartoon glow and pairing rule G-C reinforce the complementary base pairing concept. DNA polymerase as a builder. DNA polymerase is shown as a construction worker adding nucleotides to the template strand. DNA polymerase extends the strand. This introduces the enzyme responsible for synthesis. The 5-to-3 synthesis direction is shown with arrows, helping students remember that polymerase works only in one direction. The leading strand forms in a smooth, uninterrupted manner toward the replication fork. Text: leading strand, continuous synthesis. Lagging strand and Okazaki fragments. Lagging strand forming in pieces. Unlike the leading strand, the lagging strand grows in short fragments away from the fork. Lagging strand discontinuous synthesis. Floating DNA fragments. Okazaki fragments are small DNA segments waiting to be joined. Showing them floating makes it easy to visualize. Primase lays a short RNA primer on the template strand to start synthesis. Primase adds RNA primer. The primer is highlighted as a small colored block at the start of a fragment. Text: RNA primer starting point. Polymerase attaches to primer. DNA polymerase begins adding complementary nucleotides from the primer. Supporting enzymes and the stability. Single-strand binding proteins, SSB proteins, hold the strands apart, preventing re-annealing. Topoisomerase untangling DNA. DNA ahead of the fork can get supercoiled. Topoisomerase relaxes these twists. DNA supercoiling. Twisting is shown visually as a problem the enzyme solves. Replication bubble expands. As synthesis continues, a bubble forms at the origin with two forks at each end. Two replication forks move opposite. So DNA replication is bidirectional. Cartoon arrows show forks moving in opposite directions. Leading and lagging strand completion. Leading strand nearly complete. The leading strand extends smoothly and is almost done. Lagging strand fragments almost connected. Okazaki fragments are still unjoined. Ligase seals fragments. Ligase glues Okazaki fragments into a continuous strand. Primer removal. RNA primers are removed by polymerase or exonuclease. Gap filled with DNA. Gaps left by primer removal are filled with DNA bases. Proofreading polymerase. Polymerase checks for mistakes, removing wrong nucleotides. Wrong base replaced. Any mismatched bases corrected. New leading strand formed. The fully synthesized leading strand is highlighted. New lagging strand formed. The lagging strand is completed after ligase action. Parental versus new strands shows semi-conservative nature. Each new DNA molecule has one old and one new strand. Completion of replication. Two identical DNA molecules side by side. Replicated DNA molecules are identical. Zoomed out nucleus showing replicated DNA. The entire nucleus is shown with duplicated DNA. DNA ready for mitosis. DNA condenses to prepare for cell division. Circular bacterial DNA replication. Prokaryotic DNA replicates from a single origin. Multiple origins in eukaryotic DNA. Long eukaryotic chromosomes replicate from many origins. Energy molecules near enzymes. ATP is required for helicase and ligase function. Energy is required. Helicase moving forward. The cartoon helicase moves along the DNA continuously. Polymerase adding nucleotides. The new strand grows rapidly. Complementary base pairing maintained. AT and GC pairing is strictly followed. Replication fork glowing highlights the active replication site. Okazaki fragments and enzyme coordination. Lagging strand shows multiple primers. Each Okazaki fragment starts with a primer. Fragments waiting to join. Okazaki fragments float before ligase action. Ligase joins fragments. Visual glue binds all fragments. Replication bubble with two forks shows simultaneous bidirectional replication. Template strand guides synthesis. The original strand acts as a guide. Hydrogen bond formation. New complementary bases form hydrogen bonds. Parental strands visible. Old strands highlighted for clarity. New strands elongating. New DNA chains lengthen. Replication inside nucleus shows replication in cell context. Enzymes working together. Helicase, polymerase, ligase, primase coordinate. Finalization and success. Final quality check. Polymerase proofreads final strands. Daughter DNA molecules identical. Two new DNA molecules side by side. Cell ready for the next cycle. DNA is complete. S phase is done. Timeline of S phase replication. Step by step. Progress shown. Comparison. Original versus replicated. Visual comparisons shows the accuracy. Original versus replicated. DNA recalls into stable double helix. DNA waving happily. Celebratory visual text is replication success. All enzymes and steps together. DNA replication summary. The cartoon shows start to finish replication. Replication flow overview. Entire replication process complete. Mission accomplished. DNA copied perfectly. So for the next bio session, you can subscribe to this channel.